Thank you for giving me the opportunity to present. I was going to talk to you about some of the work that I've been doing as part of my PhD, which is entitled Chlamydial Epidemiology and Therapeutics in a Southeast Queensland Koala Population. So most of you would be aware that koalas are listed as vulnerable in Queensland, New South Wales and ACT, and that's because koalas in the northern part of their range, so the green area here on the map, have been suffering severe declines of up to 80% over the last 2 decades. And as a result, population management programs are going to be necessary in these populations if we're going to reverse the extinction trajectory and prevent localised extinctions. And the main threats that are driving these declines are habitat loss and climate change predominantly, but also trauma from dogs and cars. And as habitats become more fragmented, things like disease become more important, and that's mostly chlamydial. So koalas are infected by 2 species of chlamydia, but chlamydia pecorum is both more prevalent and pathogenic. And these infections are endemic in almost all koala populations. And generally, chlamydial infections have been considered to be characterised by chronic or asymptomatic carriage. But in susceptible koalas, we do see that they develop this inflammatory and fibrotic disease. So in the pictures up the top here, in the eyes, we commonly see keratoconjunctivitis, and if that's severe, that can lead to blindness. In the urogenital tract is the other site that these infections cause disease, and in the bladder this causes inflammation of the bladder wall, which we see as this urine standing on the rump due to incontinence. If that infection ascends into the kidneys, we get pyelonephritis and renal failure. If it ascends into the reproductive tract, we get these bursal cysts, these black fluid-filled areas that form around the ovaries, and they render the female koalas infertile. And in severe cases, we do also see mortality from chlamydial disease. But unfortunately, treatment of chlamydial disease can be really challenging in koalas, and that's partly because antimicrobial use can be fatal because the koalas have their specialised gastrointestinal microbiome. So we're really limited in the drugs that are safe for use in koalas. And also because the cases that often are presented for treatment are chronic cases, they've often got a lot of fibrosis and scarring, so they can respond quite poorly to treatment. So obviously chlamydial disease has these quite important impacts at the individual level. This really has bad animal welfare outcomes, these really painful conditions, but it can also negatively impact population viability in these declining koala populations. So chlamydial disease control should be incorporated into population management programs in these at-risk populations. And for the efficiency and success of this management intervention, we really need to have a thorough understanding of chlamydial epidemiology and disease dynamics and a cost-effective best practice treatment methodology. Comprehensive longitudinal population studies can provide these, but they're currently lacking due to the costs associated with these studies. And a lot of our current understanding is based on biased hospital or necropsy data sets, opportunistic or cross-sectional field studies, or studies that lack sensitive diagnostic methodologies. So instead we utilise a unique approach where we use data from a large-scale koala population management program that was conducted in the Moreton Bay region of southeast Queensland and included 503 koalas over a period of 4 years. Those koalas underwent regular field monitoring, capture and comprehensive clinical examinations by vets. And during these examinations, swabs were collected for molecular diagnostics, and also diseased koalas were admitted for treatment of their chlamydial disease. So by analysing the clinical and field records compiled during the management program, as well as the swabs using chlamydia pecorum specific qPCR and on-page genotyping to investigate chlamydial strains, that allowed us to identify and follow chlamydial infections over time. In that way, we could establish really well-defined clinical groups so that we could look at some of the factors that are important for chlamydial disease progression. And it also allowed us to evaluate some of the treatment regimens that were employed during the population management program so that we could look at the factors that were important for the short and long-term clinical outcomes, both during treatment and after treatment. So we screened almost 150 koalas over 480 individual time points, and we detected 38 new chlamydial infections. So these were healthy koalas that went from being qPCR negative to positive. And actually when we followed them over time, we found that almost 2/3 of those infections did actually progress through to disease. So in 50% of those cases, infection and disease were detected at the same time point. And in the remaining 16% of the infection was present for 1 or more consecutive clinical examinations before the koalas progressed through to disease. And in contrast to our current understanding, only 5% of the infections remained chronic and asymptomatic for more than 2 years of monitoring. And interestingly, we actually found that 29% of the koalas were able to resolve their clinical infections, so became qPCR negative without progressing through to disease. And when we used a Chlamydia pecorum specific qPCR to look at the infection load dynamics in the urogenital tract, we found that disease progression at this anatomical site was associated with the infection load. So koalas had significantly higher infection loads if they progressed through to disease compared to koalas that didn't. We also found that infection loads were significantly higher if they had infection and disease detected at the same time point in comparison to the group of koalas that were chronically infected before they progressed through to disease, or that were chronic and asymptomatically infected or were able to resolve their infections. And similarly, we found that koalas that had recently acquired infections, so if they'd acquired an infection in the last 3 months, they had significantly higher infection loads than if an infection was present for more than 3 months. Then when we looked at using on-page genotyping to investigate the chlamydial strains in the population, we found that we detected a range of on-page genotypes, but we found that on-page genotype E' was the most prevalent in this population. And in contrast to the urogenital tract site where we found a range of on-page genotypes present, we only detected on-page genotype G and E' at the ocular site, suggesting there might be some tissue tropisms associated with these strains in koalas. And we also found that certain strains were associated with disease progression in different anatomical sites, suggesting that some strains might be more pathogenic. So in the urogenital tract site, on-page genotype infections that were typed as strain F and F' were significantly more likely to progress through to disease. And in the ocular sites and the eyes, on-page genotype E' infections were significantly more likely to progress through to disease. And actually both these strains at those anatomical sites produce infection loads that are a log higher than other genotypes. We also found that resolved infections were significantly less likely to be urogenital infections in comparison to ocular infections. When we looked at the strains in some of these long-term infections, we found that we're actually detecting genetically distinct on-page genotypes over time in some cases. And this might suggest that either the koalas are repeatedly resolving their infections before they're becoming reinfected with a novel genotype, or instead, which is probably more likely, it might be reflecting the chlamydial strain dynamics in mixed genotype infections. When we looked at koalas that actually developed severe disease, so this was disease that resulted in the death or mortality of the koala, or euthanasia, or would have without veterinary intervention, which was about 10% of the cases of disease, we actually found that in these cases it didn't, disease progression didn't appear to be associated with the infection load in the chlamydial strain. And in these cases, it was prior chlamydial exposure that was associated with disease progression, and that suggests that potentially there's something associated with the host immune response that's resulting in these cases. In contrast, though, with female infertility, chronic chlamydial exposure wasn't a necessary driver, so 24% of the female koalas that developed female reproductive tract disease acquired their infections shortly after sexual maturity, so less than 2.5 years of age and potentially their first mating. And unfortunately, we found that successful antimicrobial treatment didn't appear to provide any long-term protection for these koalas. So after they were being released back into the wild, they were being reinfected less than 4 months after release and developing another bout of disease less than 5 months after they'd been treated. We found by looking at these treatment regimens that were employed during the koala management program, we were able to identify a treatment course that was almost half the previously published treatment length, but was still effective and safe. So a 14 to 28 day course of chloramphenicol still achieved microbiological cure and resulted in the release of more than 94% of the koalas back out into the wild. We found that the shorter antimicrobial course in combination with the surgical treatment of ovarian hysterectomy was beneficial for female koalas that were infertile as a result of this bilateral reproductive tract disease, and they remained healthy for a median of 466 days of post-treatment monitoring. And we found that supportive care was really important for successful treatment outcomes, and particularly the combination of oral transformation and fluid therapy was really beneficial for cases of oxalate nephrosis that had developed as a result of antimicrobial use in koalas. So overall, the research, or this research as part of my PhD, really highlights the importance of access to point-of-care molecular diagnostics and an effective koala chlamydia vaccine, so that we can identify and prevent these chlamydial infections, because more progress through to disease than we'd previously realised, and we don't need chronic chlamydial exposure to get disease progression. We identified a group of koalas that appeared to be resistant to chlamydial disease progression that warrants further investigation. We've provided a best practice treatment methodology that not only minimised treatment costs, but also the risks associated with treatment in captivity, and that obviously improves koala welfare. But also this research has contributed to successful conservation outcomes by informing management practices and also maximising the benefit of invested resources. So I'd just like to thank all of my funding bodies and my supervisory panel at USC, and a couple of other people there on the list that you'll be able to read. So thank you all for listening, and if you've got any questions, ask away.
Thank you, Amy, for a fantastic talk. We're going to have to move things a bit faster, so can we have 1 or 2 quick questions and then we'll go on? Anyone has a specific question for Amy? You're all being good citizens and sparing her the... No? Okay, all right. In that case, thanks a lot, Amy. And we'll move on to, oops, where am I on the list? To Rachele Wilson, please.
Hi. Okay, I'll just share my screen. And there we go. Can everyone hear me and see my slides? Yep, okay, great. Oh, I'll just move this somewhere. I'm not really sure how to do that.
You can't see it.
You can't see that? Okay, good, good. Okay, all right. Okay, good afternoon, everyone. I'm Rachele Wilson, and today I'm going to talk to you about a study that forms part of my doctoral research, my first chapter. And these are my collaborators here. You'll know some of them, like Alison Shapcott and Helen Wallace, and then the others are not in Australia, they're in Germany, but I'll thank them later. Okay, so I'm going to talk to you about this research which looks into the pollen diets of Australian native bees. So as we all know, bees are really important, and particularly in agriculture. So we know bees and other animals are responsible for pollinating most of the world's flowering plants, and in particular our cultivated fruit, seed and oil crops and vegetable crops. But ironically, agriculture is also one of the main reasons why we're seeing pollinator decline. So wild and managed bees are particularly sensitive to the loss of floral diversity. And we're seeing a lot of that loss happening from land use change, and agricultural expansion is the largest contributor to land use change around the world. So this conversion of natural land to agricultural land is a major threat to bee health and also pollination services. So there's lots of suggestions or studies on how we can support wild and managed bees through this phenomenon. And it might be through retaining natural vegetation, or having a wide variety and lots of different species of crops on farm, and actually going out and revegetating and planting particular things that we think they might go to for food and nesting. But you really need to understand the different requirements of different bee species, and there's lots of different bee species, and they all have different foraging distances and things like that. So you really need to understand this for each species. But what we do know is that when you have perennial colonies, so hives of social bees, in these sorts of landscapes where they might not be very diverse, you need to make sure that they have access to something year round, not when your target crop is flowering. So there's been a little bit of work done for this, particularly in temperate places and in Europe and in the US, and mostly for honeybees and bumblebees. There's not much known for our Australian bees. So I won't go into them too much because Fraser showed you some really good pictures of hives before, but just in a nutshell, there's lots of different stingless bees. They're pretty much in the tropical, subtropical areas of the world. We've got 14 species in Australia, and Tetragona carbonaria, or the sugarbag bee, is the one that we most commonly use here in southeast Queensland. So previous research from my study group looked at the colony fitness and reproduction of this species in natural and agricultural landscapes, and they found that the colonies in agricultural landscapes tend not to reproduce well. You tend to lose hives because they actually starve if they're left there too long and they don't have enough plant diversity in the landscape. But conversely, you also find that they reproduce really well in places that have lots of resources all the time, like gardens. So what we wanted to know was what plants, if you're going to go down that strategy of planting things to support bees in these landscapes, what plants are required to sustain these colonies over several years and different seasons. So that's where my study comes in. So we wanted to identify the long-term seasonal variability in pollen resources used by this species in different landscapes. And we wanted to know how does the diversity of those pollens that they forage on differ with land use and season, and what actually are those plant sources of those pollens. So we did this by installing, well, Benny Kaluza installed hives of the species in forests and macadamia orchards a couple of years ago in 2011 at 8 different sites. So we started off with 4 hives, and then because of that study, we were looking at reproduction, we ended up with up to 32 hives on the site. So there are a lot of bees. And they also did vegetation surveys there, so 500 meters around the hives in each direction, which showed that the forest sites actually had much higher diversity, twice the plant richness compared to orchards in terms of what was growing there. So because of that, we expected to see quite big differences in the diversity of the pollen that the bees forage on between these landscapes. So then what I did is I'd go around to hives in each of those sites every season, and I'd take pollen from the hives, and then it gave us 166 samples from 57 hives. Then we would go through this process of DNA metabarcoding to identify the species that were the source plants of those hive pollens. So extract the DNA, PCR, 2 different markers, also adding molecular tags to those samples, so that way when you sequence them on the MiSeq all at once in the one library, you can bioinformatically separate out all of those sequences to figure out which sample it came from. And then what we'd do is we'd match the sequences of that pollen DNA to the sequences of the plants that we knew were growing in the area because of the surveys. And so from downloading the different barcodes that are available on GenBank, and from some of the barcoding done in-house for those that weren't barcoded, we managed to cover about 93% of the species that we knew were growing at the sites with these 2 markers. So what did we find? So this plot here shows the diversity and richness of the pollens that we found in different seasons, which are the colours, and in land uses. So we've got spring, summer, autumn, winter. And you can see it kind of goes like up, down, up, down. So we found there's seasonal differences in the richness and diversity of the pollen sources. But what we didn't find was differences between the land use. So for each of these, you've got forests on the left, orchards on the right, forest, orchard, and these are all the same, which is really interesting. I'd just like to note here that most of the diversity happens in the dry seasons, so like in autumn, sometimes in forests, sometimes in summer for forests, and it's lowest in spring, particularly in the orchards. So it's probably because they're mostly going to macadamia. So why didn't we see any differences in the land use? So if you look here, we've got ordination of the different pollen sources, and in blue are the orchards and in pink are the forests. So the reason why we're not seeing any differences is because what's available to hives in orchards is basically a subset of what's available to those in forests, but it's still quite diverse. So the hives in those orchards basically have less options to choose from, but they still manage to to collect a similarly high amount of diversity to those in the forests. So when you actually look at the identity of those species, we found over 302 species of plant sources for these hive pollens, some only to genus levels. That's why I've got 341 taxa there. And most of those species have not been previously published as forage for Chlamydial carbonara before, and 37 of those families haven't been reported for stingless bees at all in the literature so far. But interestingly, of the families that we did find, a lot of them, like a lot of the most common ones, were also common to studies of stingless bees in other parts of the world. So particularly Proteaceae, Asteraceae, Myrtaceae, Araliaceae. So the question then is, are these plant families attractive to stingless bees, or are they just plants that seem to be quite widely distributed in the tropics and subtropics? So that's just what they happen to go to. We don't know that, but it's something to look into later. So when you actually look at the sources that we found in the orchards, this is just showing the proportion of pollen sequences for each of the species we identified in spring, summer, autumn and winter. And on the left, we've got one marker, on the right, we've got another marker. So don't worry if you can't read the species yet, because I'll go into that later. But the key thing here is that you see that the source plants vary with the seasons. So you can see in spring, the reason why that diversity is so low is because they're going heaps to macadamia and then a little bit to these weeds. And then you want to see, if you look here in autumn, there's lots of bars because they're going to lots of different things. So that's when diversity peaks. Another thing that's interesting is that a lot of these sources are native trees and shrubs, but also grasses and sedges, which was really weird. We didn't expect that. But looking at the literature, there's actually been a really good review that came out recently that shows that bees do go to grasses and sedges, and we think it might be an important source of protein, particularly in the dry season. But also introduced plants. So of course we'd expect to find this in the orchards. There's a lot of weeds of agriculture there, but it was just interesting to see how they use them over the seasons. But the main take-home message here is that what we're seeing is many small and few large sources, except for that macadamia. Like most of these are less than like 0.1, so less than 10% of their diet is coming from lots of different species. In the forest, it was a really similar story. They have access, except for that they have access to those native trees and shrubs and more species of native trees and shrubs year round. They tend to be going to some things in every season. And that's probably, it might be because in the orchards, they just couldn't be bothered flying that far to go to, you know, those strips of native vegetation because they already had lots of macadamia or something else. And interestingly, we still see those grasses and sedges. So it's not necessarily that they're going to grasses because in orchards there's nothing there. They might also just be attracted to them, particularly in that dry season. And even though it's forest, we're still seeing those introduced plants. So that's really interesting. It's like, are they helping them to, well, they're probably obviously helping them to reproduce and spread, but also maybe introduced plants are so good at reproducing and spreading because they're very attractive to the things that help them do that. So lots of questions. So in summary, we found that high pollen diversity didn't differ between the land uses, despite what we expected based on the plant surveys. And we think this might be because colonies and orchards flew further to forage on a range of things to bring up that diversity, or when they were, for example, in spring mostly foraging on macadamia, they would also mix that with weeds and things that are growing close to the hives. But interestingly, of those plants that they went to, only half of them they're only going to about half of what's identified in the surveys. So there's a lot more out there that they're not going to. And what they're going to isn't always the largest, most prolific species. They're not always the most common. So this suggests that what they're going to is probably driven by preferences like flower morphology, like the color and the shape, or the chemistry or the nutritional quality of that pollen. So there's some more research there to look into what it is that's driving those preferences. And yeah, most importantly, what we found was many small and few large pollen sources. So most of the plants that you wouldn't have been able to see on the plot because they wouldn't even show up were less than 1% of their diets on average. There were only about 15 that were greater than that. And this is really similar to a study that came out recently for honeybees, which found really similar things, only about 10 taxa at greater than 1%, even though they had a really, really wide variety of flowering plants available at the time. So we think this means that when these social bees are in landscapes with high floral diversity, they're kind of lazy. They think, oh, we've got so many options to choose from. Let's just go to, you know, the closest thing that gives us the most nutritional benefits. So we think that they're optimizing their foraging using fewer resources. So what does this mean in terms of management of social bees, not just stingless bees? So this shows that bee colonies can compensate in resource poor landscapes by varying their foraging behaviour, and it looks like they have clear preferences for particular species. We know that bees aim for a diverse, nutritionally balanced pollen diet to ensure their colony performance. So that means that if you want to help them to have that diverse diet, you need to make sure you have diverse range of floral resources on farm. And this might even include just those introduced weeds that people would normally mow between the rows, and keeping some of those, even the wind pollinated, like the grasses and things, which could provide critical nutrients for bees during periods of poor pollen availability. So in a nutshell, two different ways in which people can do this is through targeted plantings, so of the species, which I'll show in the next few slides, or of probably members of these families, which we found highly abundant in their diets, but also in the diets of other stingless bees. So not just necessarily in Australia, but this could be applicable to those in other parts of the world. And also through managing existing vegetation. So like I said, those grasses and sedges, which are probably in riparian zones, which most farms should have access to, like creeks or dams, and keeping those weeds between the rows, but particularly the annuals and things that aren't declared weeds. So stuff that's only going to pop up for a short amount of time, like this billy goat weed, which you can see a bee on here. So if you're interested in this sort of thing, these are the top 20 species that we identified in those samples. And you can break them down into the different seasons and their indigeneity. So if you're a bit of a purist and you only want the natives, then you can just plant those. But if you want to know which weeds to keep, that blue billy goat weed, wild radish, and guinea grass, most of them are annuals. And so in my opinion, I don't think they're really too dangerous to keep as introduced plants, except for that guinea grass, but that's just, it's planted as forage for cows and stuff, so I'm not too worried about that. And most of you will probably recognise that what's it called, the piccabean palm. Piccabean palm, they love that in autumn. And of course, keep all your eucalypts and corymbias and things. But if you want a copy of that, you can always email me there. Yeah, so that's it. I'd just like to thank obviously my supervisors, Helen and Alison and Sarah, and Abigail and the GeneCology Research Centre for your support, and all the people who provided us grants and scholarships. This isn't published yet, but it's in review now. So, but if you have any further questions, just feel free to email me. Thank you.
Thank you, Rachel. That's lovely work. I mean, we're all aware that bees are so important for our ecology and our future, and it's really good to know that there are ways to help them. I also want to acknowledge the really pioneering work that you were doing and Alison's group were doing on the barcoding way before other people were doing it, and I know you came across you had to sort out a lot of issues. So thank you for persisting and doing it great. We're awfully late in time, but is there a quick question for Rachel from anyone? Okay, Lockie asks, how did you estimate the abundance?
Yeah, so
really quickly, yeah.
Very quickly, yeah, it's relative abundance because we know that you can get biases with PCR. So it's basically based on, yeah, the number of copies of those sequences that you get for each species, but relative. So that way you can control for the differences that you would get, yeah, with PCR.
I think, Lockie, you might need to have a coffee date with Rachel to discuss.
Yeah, no worries.
Thank you so much. And we now move